I'm super curious but pretty skeptical about peptides. I hear a lot of influencers talking about them like they're this magic cure-all snake oil. I think if you are interested in these, it's really important that you know what you're getting involved with. So I'm doing a bunch of research and breaking down the studies so you don't have to. Let's get into it. Today we're talking about Epitalon. So your chromosomes have little protective caps on them, and every time your cells divide, those caps get shorter. When they're gone, the cell stops dividing. Now your body has an enzyme that rebuilds them. In a lot of us, though, it's just barely turned on. These caps are called telomeres, and they sit at the ends of your chromosomes and protect your DNA during cell division. Every time a cell divides, they shrink. Your body also makes this enzyme called telomerase. Its job is to rebuild those caps, but in most adult cells, telomerase is almost silent. Here's where it gets wild. Since the 1960s, scientists have known that human cells can only divide a set number of times. It's called the Hayflick limit, and it's one of the most basic rules of cell aging. When telomeres get too short, the cell shuts down. It stops dividing, it's done. So they asked, what happens if we wake up telomerase? Researchers tested a 4-amino acid peptide called Epitalon. They added it to human fibroblast cells in culture. Now these are connective tissue cells, and they tracked them through dozens of rounds of division. The control cells stopped dividing at passage 34. That was their Hayflick limit. Their telomeres had shortened to the point where division just stopped. But the cells that got Epitalon, they kept going. They hit passage 44, 10 extra divisions, and they were still dividing when the study ended. Think of it like this. Epitalon didn't just slow the countdown, it reversed it. Telomeres in the aged control cells shrunk well before early levels, so cell division stopped. But in the treated cells, telomere length came back. It returned to the same size as young, early passage cells. The peptide activated telomerase. The enzyme rebuilt the caps. And the cells pushed right past their supposed Hayflick limit. Now, this was a cell culture study, not a study in living humans. Cells in a dish don't behave the same way as cells in your body. So we cannot say for sure this works the same way inside of you. More research is needed. But your cells already carry these caps, and your DNA already codes for the enzyme that rebuilds them. The system exists. What if we're just learning how to turn it back